In this chapter, we looked at factors that can influence the outcome of the phenotypes in Mendelian crosses. Here is the outline of the chapter, and for exam 1 we covered until 5.3. We did not cover anticipation or environmental factors. Multiple alleles are different forms of one gene that are present in a population. Here as example, it was given the ABO blood group, and you know that a person can be type A or type AB or type O, but a person cannot be ABO at the same time. For each particular locus, you can have 1 or 2 alleles, but you know that the population has more than 2 alleles that can be expressed. And here is the definition of an allele. These are 2 versions of the same gene, and each version of this gene is defined as an allele. They are basically identical, but with few DNA sequence variations. The important thing to know about the definition of an allele is that it is just different variation of the same gene. Now, when we started to look at the extensions of the Mendel principles, we started looking at different types of dominance. So here we saw incomplete dominance. This type of trait would appear more like the blending principle that we had seen before. So here, if you have, for example, one flower that is white and you cross with one flower that is red, you do not get a white or a red, but you get something more or less in the middle, which is this pink flower. And the important thing to remember here is that this type of dominance affects the phenotype, but not the way in which the genes are segregated and inherited. The principle of dominance says that the dominant allele will mask the effect on the phenotype of the recessive allele. Now, when you have codominance, you have both alleles that are being expressed, and neither of them backs down, meaning that you see both of the phenotypes. The example given was of sickle cell anemia and also the ABO blood group. And I also mentioned in class the case of cystic fibrosis.
Sickle cell disease is a blood disorder that is inherited from both parents. This disorder is more common in certain ethnic groups, such as African Americans, Arabs, Greeks, Italians, Latin, and Native Americans. Normal red blood cells are very flexible and are circular in shape, resembling a donut. Their flexibility and shape allow them to travel freely through small blood vessels called capillaries. In persons with sickle cell disease, the red blood cells become crescent or sickle-shaped and also become inflexible. The abnormal cells stick inside the capillaries, blocking blood flow to vital organs. Persons with sickle cell anemia can have symptoms such as yellow-appearing eyes and skin, pale skin, delayed growth, bone and joint pain, increased risk for infections, development of leg ulcers, eye damage, anemia, and damage to the organs affected by the obstruction.
In the case of cystic fibrosis, I had mentioned that the type of dominance would depend on the level of the phenotype that you're looking at, which means if you look at the receptors, transmembrane channels at the molecular level on a person that is heterozygous for the alleles, you'll see that this person will express both types of channels, one that is defective on the transporter of chlorine ions and the other one that works properly. So at the molecular level, you'll see the case of codominance in which both channels are expressed. But when you look at the physiological level, you do not see codominance, you see the dominance effect or the dominant phenotype of the normal chlorine transport channel. So at the physiological level, it appears that the mutated channel is recessive. Cystic fibrosis is an autosomal recessive genetic disorder. And what that means is that it's in an autosomal chromosome, not in one of the sex chromosomes. Still, this disease, when inherited as a homozygous recessive, has a very low life expectancy. Here we see a normal channel that is capable of opening and exchange chloride ions between the inside and outside of the cell. And mutations on that channel, the CFTR channel, will cause the channel to not work properly, and that will cause the symptoms of cystic fibrosis. There we have the channel that is working properly. Now you block the movement of salt. Knowing the genetic characteristics of cystic fibrosis is important, but I also think it's very important to see that this disease is affecting real people.
My name is Violet and I have cystic fibrosis.
What is this?
On my stuff.
Your stuff for what?
What's this? Listen to this.
Do you know what those are?
My weight settle.
Your drinks? The powder for your drinks? Okay.
It's big one for my straws.
You drink them out of your straw. When do you have them? At dinner? Do you have them at breakfast and lunch also? And snack? Every time you eat, you have to take those.
We have looked at the case of dominance, where one of two alleles will mask the effect of the recessive allele. So in this case, if you have the Punnett square and a cross of two heterozygous, we are going to have a 3-to-1 ratio of dominant over recessive phenotypes. Now, in incomplete dominance, the phenotype of the heterozygous is different than in complete dominance. In incomplete dominance, you have a ratio that is 1 to 2 to 1, meaning that 1 will show the phenotype of the one of the dominant alleles, and 2 are going to be a new phenotype that is intermediate between the two dominant phenotypes. Now, in codominance, you have both alleles that are expressed, as it was the case in the heterozygous form of cystic fibrosis at the cellular level, and also in the case of the AB blood group.